Now we will continue with the topic. Uh, in this part, I'll be covering umbilical cord along with the twinning. So, what is umbilical cord? By definition, it is a tubular cord-like structure by which fetus is connected with the placenta. So, it is covered by amniotic membrane. So, during the lateral folding, if you remember, the amniotic cavity overgrows whole of the fetus. It uh, covers the ventral surface as well, and it comes to cover this connecting stalk as well. So that is why it is covered by the amniotic membrane. So now it has two ends. One is the placental end, another one which is attached to the center of the placenta, and another one is the fetal end, which is attached to the umbilicus of the fetus. So in a full term baby, the umbilical cord's length is around 50 to 55 centimeters, and it is around 2 centimeters thick. Placenta was 3 centimeters thick, it is 2 centimeters thick. Then how it is formed? So on the formation of the embryonic folds, the yolk sac and the connecting stalk, they come to lie on the ventral surface. So this is the lateral folding, cephalocaudal folding going on. This one is the connecting stalk. This was the part in the bilaminar or the trilaminar germ disc where the extra embryonic coelom was not intervening between the cytotrophoblast and the somatopleuric membrane. So it is devoid of the chorionic cavity. So this connecting stalk will form the future umbilical cord. So later on there was allantois formation also, which was there. So on the formation of embryonic folds, the yolk sac, this was the yolk sac, which was also taken inside by the embryo because of the cephalocaudal folding and the two lateral folds. And the connecting stalk, it comes to lie on the ventrals. Because of this fold, the connecting stalk and the yolk sac, they will come to lie on the ventral surface of the fetus because of the cephalocaudal folding. So the and also because of this folding, the amniotic cavity is encircling the whole of the fetus. Over here you can see, so amnio-ectodermal junction comes to lie on the ventral surface of the fetus, and it forms a umbilical ring through which vitello-intestinal duct, if you remember, this communicates the umbilical vesicle with the midgut, and the allantois protrude. So this the umbilical ring, it is protruded by the umbilical vesicle or the vitellointestinal duct and the allantois. Now the extra embryonic mesodermal tissue of the connecting stalk, it also gets vascularized to form the two umbilical arteries and two umbilical veins during third and fourth week of intrauterine life. Now, during the later part, the right umbilical vein gets obliterated and only the left umbilical vein is left. The mesodermal connective tissue of the connecting stalk forms the mucoid tissue, which is known as Wharton's jelly. So, it is the only form of embryonic tissue which is present in adults. Adults, I mean, in human beings after birth, not in adults, but in human beings after birth. So what is the contents of umbilical cord? So it has got a Wharton's jelly, two umbilical arteries and a umbilical veins. So the umbilical arteries and veins come under umbilical vessels. So there are two umbilical arteries and one umbilical vein. Left is left, right will disappear. Umbilical arteries, they will carry deoxygenated blood from internal iliac arteries of the fetus to the placenta. Umbilical veins are two in number, but right will disappear. So left will carry oxygenated blood from the placenta to the fetus. Left umbilical vein will join a left branch of the portal vein that convey the blood to the inferior vena cava via ductus venosus. This we will study in detail. Then what is Wharton's jelly? It is a mucoid connective tissue of the umbilical cord, which is also known as Watten's jelly. So twisting of the umbilical vessels produces beaded appearance of the umbilical cord. Now, other contents are the part of allantoic diverticulum. If you remember, allantois was the outpouching of the a part of the yolk sac, the tail part of the yolk sac into the connecting stalk. So, part of the allantoic diverticulum enters in the umbilical cord and it undergo fibrosis to form urachus. And remaining part of allantois form a part of urinary bladder. Then the vitellointestinal duct and umbilical vesicle, the part of the yolk sac communicating with the midgut, forms vitellointestinal duct. Unabsorbed part of the yolk sac forms umbilical vesicle. In later part of pregnancy, vitellointestinal duct and umbilical vesicle, they will disappear. So the components of umbilical cord are two umbilical arteries, one umbilical vein, Barton's jelly, part of allantoic diverticulum which will form urachus, the vitellointestinal duct and the umbilical vesicle both will be absorbed after delivery. Then is amnion. What is amnion? It is a thin membrane that lines the roof of the amniotic cavity. So inner cell mass during second week of the trophoblast, inner cell mass of the embryoblast will form an amniotic cavity and get separated from a layer of amniotic cells. Then amnion forms roof and bilaminar germ disc forms floor of the amniotic cavity. So amnion consists of angiogenic cells covered by somatopleuric layer of extra embryonic mesoderm. So by the end of 8th week on the formation of embryonic folding, amnion entirely covers the embryo. So amniotic cavity, it contains amniotic fluid. It is also known as liquor amni or camerous fluid. So what are the constituents of amniotic fluid? Water, electrolytes, fetal wastage including urine, human chorionic gonadotropin, human placental lactogen, cells exfoliated from fetus. Now, what are the functions of amniotic fluid? It helps in shock absorption. So it provides water cushion that absorbs jerks and protects the fetus. It gives nutrition to developing neuroectoderm as it passes through anterior and the posterior neuropore. Then it allows free fetal movements and the fetus excrete urine in amniotic fluid through allantois. Fetus swallow and absorb amniotic fluid through the gut into the fetal blood. Finally, fetal waste products reach maternal blood through fetoplacental circulation. Then by determining the amniotic fluid volume through sonography, we can assume the gestational or we can make out the gestational age. So by 10th week, it is around 25 ml. By 20th week, it is around 400 ml. At 28th week, it should be around 800 ml. By 36th week, it should be 1 liter. And again, it started decreasing that by 42nd week, it becomes 400 ml. So it is maximum at birth at around 36th week, that is around 1 liter. So its volume increases from 10th week to 28th week and then it starts decreasing. Now, what are the clinical aspects? One is amniocentesis, that is extracting certain cells from the amniotic fluid, and we can uh, subject those cells for biochemical analysis to detect some inborn errors of metabolism, as well as we can subject those cells for karyotyping for determination of certain genetic diseases. Then oligohydramnios is production of lower amount of amniotic fluid than normal. Then is amnion nodosum. Then is Potter's syndrome. Then is excessive production of the amniotic fluid, that is polyhydramnios. So over here you can see there is limb constriction. which is probably secondary to amniotic bands formation. So because of the domestic violence, there can be loss of some of the amniotic fluid, which may produce certain bands around some developing fetal area. So it may lead to constriction of limbs whenever those amniotic bands will be forming. So it is a result of oligohydramnios. Then is a condition known as Hydrops Fetalis. It is a condition which is characterized by accumulation of fluid or edema in fetus. Location of Hydrops Fetalis includes subcutaneous tissue, that is scalp, the pleural cavity, the pericardial cavity, and the peritoneal cavity. The fetal anemia is most common cause of Hydrops Fetalis. In the above image, the protruding abdomen due to ascites and edematous face is seen. So mainly whenever there is a blood antigen incompatibility, this condition is mainly seen. Then is amniotic band syndrome, a right lower limb showing constriction rings in lower part of the leg and at proximal phalanx. Over here you can see. Then left hand, it is showing constriction ring at the middle phalanx. Then left foot is showing constriction band at the distal phalanx of the great toe with absence of nail. So all these are amniotic band syndrome. They will form constriction rings around the certain areas, and after the birth, those will come out as certain developmental anomalies. Then is the fetal membranes in twins. So there are two types of twins, monozygotic and dizygotic twins. The two-thirds of the twins are dizygotic or fraternal twins. They result from simultaneous shedding of two oocytes and fertilization by different spermatozoa. So because there is formation of two zygotes, and so totally different genetic constitutions of the twins will be there, and they might be of same or different sexes, and resemblance will not be there. Then the zygotes implant individually in the uterus. So they have their own placenta, own amnion, and own chorion. So they are also known as dichorionic, diamniotic, and dizygotic twins. So this is a fraternal, two different zygotes are being formed, and so they have their own embryoblast, two different embryoblasts with the two different trophoblasts. So two different placenta with the two chorionic cavities. So they will be dichorionic, and since there will be two embryonic discs, so there will be two amniotic cavities will be forming. So it will be diamniotic, dichorionic twins, dizygotic twins. Then is monozygotic twins. So here the rate of monozygotic twins is 3 to 4 per 1000. It results from the splitting of the zygote at various stages of the development only during cleavage. जब cleavage चल रहा है तभी division हो जाए. So the earliest separation is believed to occur at the two cell stage. Two cell stage पे अगर division हो जाए, the cells are totipotent. So blastocyst will be formed, two blastocysts will be formed, क्योंकि embryoblast भी दो बनेंगे, trophoblast भी दो बनेंगे. So the blastocyst implants separately, and each embryo has its own placenta, own chorionic sac. So monozygotic, but since the zygote is one, so monozygotic, dichorionic, and diamniotic. ठीक है. So how they differ from dizygotic? The both sex will be same since they are forming from a common zygote. So they will have close resemblance, and they too can be recognized as partners of monozygotic pair by strong resemblance in blood groups, fingerprints, sex, and external appearance such as eye and hair color. So everything will be similar, though they will be having different placenta, different chorionic cavity, and different amniotic cavities. So over here, this is a different types of monozygotic twins. So here the early division has taken at the 2 cell stage, which is giving rise to monozygotic dichorionic diamniotic twins. The second is the division occurring in the inner cell mass before the formation of bilaminar germ disc. So now, because the embryo blast is being divided, so there will be two amniotic cavity, but since the trophoblast was same, so a common chorionic cavity. So monochorionic and diamniotic monozygotic twins, right? And over here, the bilaminar germ disc is formed. Just before the formation of primitive streak, there will be a division. So what will happen? They have a common chorionic cavity since the trophoblast is common, and since the embryoblast was also common, so they have a common amniotic cavity as well. So monoamniotic, monochorionic twins. So the monochorionic diamniotic twins, the splitting of the zygote, दो amnion है यानी कि दो inner cell mass थे. तो splitting of zygote usually early at early blastocyst stage. The inner cell mass splits into two separate groups of cells within the same blastocyst cavity. The two embryos have a common placenta and a common chorionic cavity, but separate amniotic cavities. ठीक है, तो एमनियोटिक कैविटी अलग अलग है लेकिन कोरियोनिक कैविटी सेम है क्योंकि ट्रोफोब्लास्ट इज सेम. Then monoamniotic, monochorionic, when the separation occurs at the bilaminar germ disc just before the appearance of the primitive streak. This method of splitting results in the formation of two partners with a single placenta and a common chorion and a common amniotic sac. So although the twins have a common placenta, blood supply is usually well balanced. In rare cases, such twins may not fully separate, resulting in the birth of conjoined twins. Such twins are also known as Siamese twins. So when the division of this bilaminar germ disc is not complete, it may give rise to Siamese twins. So there are other defects as well, twin defects. That the term vanishing twin refers to death of one fetus. The twins होते हैं, एक fetus, one fetus if it dies, it will disappear gradually, which occurs in the first trimester or in the second trimester, and it may result in the resorption of the whole of the fetus, resulting in fetus papyraceous. Like a paper, it will it will squeeze. So fetus papyraceous. Then is twin-twin transfusion syndrome, which occur in monochorionic monozygotic pregnancies, in which the placental vascular anastomosis, which occur in balanced arrangement in most monochorionic placentas, are formed, so that one twin will receive most of the blood flow and the flow of another twin is compromised. So twin transfusion syndrome, one twin will be larger, another will be compromised. Then there are different types of conjoined or the Siamese twins. If the separation is incomplete at the thoracic region, then they are known as thoracophagous. If they are joined at the sacral region, they are known as pyophagus. And if they are joined at the cranial region, then they are known as craniophagous. That's all. This completes our general embryology. Thank you for listening.